Thank you again all for having me today. My name is Meghan Yap-Chiongco. I am a postdoc at the University of Copenhagen, but I kind of nominated the species and I'm so happy that the species Wirenia argentea has been chosen. It is one species I worked with a lot in my PhD at the University of Alabama. And so I will kind of get along to what this guy is, because I'm sure most people have not seen something like this before. Um, so believe it or not, Virenia argentea is actually a mollusk. So it is, if you can see my pointer, it is here on the molluscan tree of life. So we generally think mollusks, we think of snails and cool cephalopods and bivalves, but this belongs to this group called Aculeifera, which is sister to all of the kind of mollusks that you tend to know. So just inherently, Virenia has a very important phylogenetic position within mollusks to understand molluscan phylogeny and evolution as a whole. These are actually found in quite high abundance in the northern European Atlantic, as you can see on the first slide, in silt and mud, living on top, eating jellyfall from about 150 to 600-meter depths. And it is the closest thing in selenogasters that we have to a developmental model. The development has been tracked using light microscopy, electron microscopy, and confocal laser microscopy, as well as some in situ hybridization. So it is kind of our developmental model that we can use for comparison within this group. Currently, there's low coverage genome data and some transcriptomes available, but there is no high quality genome to kind of really push this forward for use in development. So what we're going to use this for, so first off, comparative genomics of selenogasters and apocophora in general. So there are only about four apocophoran genomes in total, but this would be the first within selenogasters within this clade called Phyllodiscipia. So it is kind of sister to the rest of the selenogasters. They're relatively small bodied, while some of these other selenogasters tend to be large bodied. And we think that the ancestor of selenogasters was primarily small and maybe even paedomorphic. So this is going to be really interesting for studying shifts in life history within selenogasters, but also as kind of a general theory for shift in life history. This will also help us explore this morphological bottleneck that we see in selenogasters and what those genomic underpinnings of becoming secondarily so simplified is. So, I mean, as you can see, something that is small and worm-like, so this is only a couple millimeters in length, has lost a lot of what you kind of generally associate with a mollusk. Again, this is going to be very important for comparative molluscan genomics, such as molluscan innovation, or my favorite, biomineralization. So these actually lack a shell and instead are covered with these calcareous spines and spicules called sclerites. And so another genome from these animals are really going to help us to understand some of these processes in this really important phylum of mollusca in general by taking these kind of lesser known weirdos. But again, thank you so much, and I will leave it there.